Hey everybody, I hope that you're having a good day. I am going to go ahead and get moving. We're going to be wrapping up our discussion of lipids. In addition to that, we're going to be getting pretty deep into our discussion of membranes. And then we'll go ahead and save some of those last elements, lipid metabolism and maybe some membrane transport for right before your next exam. Okay, so lipids. When we talk about lipids, yes, I'm recording good. We've already talked about lots of the nomenclature elements of lipids. We've talked about, some of the different nomenclatures, the delta and the omega scheme. In addition to that, we've talked about the physical properties, how lipids pack really well together. In addition to that, um, they're largely hydrophobic. And whenever I say they pack really well together, that's true of both saturated and unsaturated fatty acids. And here what you can see is sorry, that's not what I wanted. Um, a mixture of your saturated and unsaturated fatty acids don't pack quite as well as a mixture, or sorry, of purely saturated fats. So those are some distinguishing factors. Now, in addition to that, individual fats and fatty acids have physical properties that vary from one another. As the number of carbons, the length of the fatty acid increases, the melting point increases. As the number of double bonds or the degree of saturation, I want you to be comfortable with that terminology, the degree of saturation, kind of the number of double bonds increases, well, then your melting point decreases. So there's a direct relationship between number of carbons and melting point. And then there's an indirect relationship between number of double bonds or degree of saturation and the melting temperature or the melting point. Okay, so here's a couple of different ones. We've got our N or omega notation described here, as well as the delta notation. Now, we talked about triglycerides and triacylglycerols. Those terms are interchangeable. The composition of saturated fatty acids in fats that you might be familiar with, olive oil, butter, and beef fat, they're going to vary a little bit. The more unsaturated fatty acids you have, the more likely that something is going to be a liquid at room temperature. The higher concentration of saturated fats and saturated fatty acids, well, the more likely something is to be a solid at room temperature. Now, triacylglycerols or triglycerides have a huge amount of energy, and they provide a huge amount of energy. I mentioned olestra was kind of a carbohydrate derived fat and fat substitute that was used in I guess you would say flavoring, because it definitely changes the way that how somebody eats something. Um, but ultimately that caused some pretty significant digestive issues. Now, there are two processes that we need to be familiar with. There's the industrial process of breaking triacylglycerols or triglycerides down, and that is known as saponification. That it's regularly used in the production of soap. Enzymatic hydrolysis, by contrast, is what your body does. Your pancreas, as well as I believe your liver, are responsible for breaking up triacylglycerols into individual fatty acids. And they're going to make them the ionized fatty acids. So you're not making a sodium salt of a fatty acid with enzymatic hydrolysis. Instead, you don't need that sodium, or that sodium's not there because you're not using sodium hydroxide in order to break it up. Now, when you talk about membranes, I think it's really important to think about, well, what's the kind of most common depiction, or what's a very common depiction of a membrane? Well, you look at a lipid bilayer, and pretty commonly you show something like this. And these two lines are not a mistake. There's only two of them, and those represent your fatty acyl or fatty acid tails. And then, so this would be your nonpolar tail. And then this at the top would be your polar head group. Now, with that in mind, if we look at something like a triacylglycerol, that is considered to be a non-polar fatty acid, or sorry, non-polar lipid, because of the fact that Yeah, glycerol has, and there is an ester bond connecting them. But if you look at the King's ransom of the atoms within that, it's a very nonpolar molecule. The hydrocarbon chains are all nonpolar. And so I'm circling our nonpolar lipids with this kind of, I guess, maroon or kind of burgundy colored um, ink. And then I'm going to circle and highlight our polar lipids using a green. Okay, so two examples are a sphingomyelin as well as a glycerophospholipid. Now, I want to show you some of the similarities and differences of these. Okay, so first of all, I'm going to highlight my glycerol backbone. Okay, so there's the three carbons that constitute my glycerol backbone of a triacylglycerol. Here's three carbons that also constitute essentially glycerol as a building block. And then it's a little bit of a stretch, but here's those same three carbons or similar, a three carbon backbone that's used for a sphingomyelin. Now I want to highlight, changing color. I'm going to go with this burgundy for nonpolar groups. Okay. So a triacylglycerol has three nonpolar groups. A um, by comparison, a glycerophospholipid has one and two nonpolar groups. Now, what make a glycerophospholipid different from a triacylglycerol is the presence of a phosphate. So if we were to look at a triacylglycerol and compare it to a glycerophospholipid, the biggest difference is going to be, well, on one of those carbons, there's a phosphate. Now that phosphate, and actually let me go ahead and extend that highlighting, that phosphate there connects a molecular group that's simply represented by X here. Now that X is a group that basically you can substitute a hydrogen for a number of different things. Now, if we look at a sphingomyelin by contrast, what we've got is something a little bit more complicated than that.
Dad.
So with a sphingomyelin, we still have that small group, that three carbon backbone, but then things get a little bit kind of haywire, really. I mean, for starters, in our sphingomyelin backbone, we've got a hydroxyl group. Okay, similar to a glycerol. However, it doesn't really, we don't have modification to it. Now, what makes this sphingomyelin similar to maybe a glycerophospholipid is we have this phosphate group. Now, that phosphate group is how we connect an X group to a sphingomyelin. But this right here is your kind of base model or your boilerplate model for a sphingomyelin. Now, in other positions, we have an amino group, and then we have a palmitate, which palmitate, or also known as palmitic acid, is a 16 carbon long fatty acid. Then, right here, on our other carbon of our glycerol backbone, we've got a 14 long well, it's a 15 long carbon-carbon chain. And for that, this is one of the reasons I'm going to do some erasing and revisit this palmitate, or sorry, the sphingomyelin. Sometimes what you will hear about a sphingomyelin is that it's an 18 carbon long chain, or it's an 18 carbon long backbone. So we've got our three. Let me go ahead and get my pen again. Three carbons here in this box. And then in this box, focusing in on these right here, that's 15 carbons. So 3 plus 15, we've got an 18 carbon long backbone there. So sometimes you might hear about sphingomyelin referred to in that context or displayed that way. It's still, however, I think valuable to group it with your lipids because one, functionally it is a lipid and structurally it is a lipid. Classification, it is absolutely a lipid. But I think that it's helpful to put it in the context or look at it in a way that's similar to a triacylglycerol or glycerophospholipid. So I always kind of think about sphingomyelin as a 18 carbon long fatty acid, or almost like an 18 carbon long glycerophospholipid with some tweaks to it. Okay. So I can clear that because what we're going to first look at here are our glycerophospholipids. And specifically, we're going to get after that X group. Now, these non-polar lipids, like what we saw with our triacylglycerols here, those are for energy storage. That's the primary function of them. And that's your best example of a non-polar lipid. Your polar lipids, by contrast, your glycerophospholipid and sphingomyelin, these are the basis for bilayers, which is one of the reasons that whenever you see a lipid bilayer, it's typically drawn with that kind of circular head and then two tails. Well, those two tails that I'm referring to would be this and this, or this and this, as is the case of a sphingomyelin. So what that means is that this right here and this right here, that's basically your polar head group. So anytime you hear about a lipid bilayer being made up of non-polar tails and polar head groups, that's what we're referencing. Now, thinking about those different glycerophospholipids, well, they are major lipid components of biological membranes. They're amphiphilic molecules with nonpolar aliphatic tails and polar phosphoryl X head groups. So amphipathic means that a molecule has a both polar region and a nonpolar region. Here's a total of 3 examples, kind of as building blocks, thinking about a glycerol 3 phosphate molecule, and then you lipidate that, or you fatty acylate that by adding fatty acids to it, and you get your glycerophospholipid. One kind of completed full molecule shown over here would be 1 stearoyl, 2 oleoyl, 3 phosphatidylcholine. Now that name is in reference to, well, carbon number 1 right here. That fatty acid is stearic acid. Carbon number 2, this is oleic acid at carbon number 2. And then that means that carbon number 3, we have this head group known as phosphatidylcholine. Or sometimes if you investigate lipids at all, you'd know this as PC. Okay, so other examples of common glycerophospholipids or common head groups are shown here. Now, one of the things that I always like to reference and highlight is serine here. Serine, this is the head group. Now, serine's an amino acid. What always throws people for a loop a little bit is to look at serine in this, kind of single line display. Some of y'all might be comfortable doing this, others might need a little bit of a helper here. So I think that it's beneficial to kind of identify, okay, what does serine look like? Serine as an amino acid looks like this. H3 plus C, that's my alpha carbon, and then my carboxylic acid here, and then here's my hydrogen. Okay, so if we look at serine here, this is that group right there. This is my carboxylic acid right there. Now, serine as a part of the molecule known as phosphatidylserine is not in a straight line like this. So it's not, you don't have something that looks like this. H, H, C, H, N, H, H. So it instead is that amino acid. I'm going to go ahead and stop that drawing just so, in the event that you weren't thinking that, nobody gets confused or anything like that. So whenever I draw serine from this, or I'm translating from this single line notation, what I've just underlined right here, now I'm putting a star underneath, that is my alpha carbon. So that alpha carbon has an amino, that NH3, that COO minus, and a hydrogen coming off of it. And then it also has CHH. And then this bond right here is what actually connects this whole thing to that oxygen right there. So another way to depict this would be, well, there's my carbon, hydrogen, hydrogen, carbon, and H3 plus HCO minus. Okay, so that's effectively what that looks like. Now, what I'd like to draw your attention to whenever I show you all of these different head groups is one, I'm not expecting you to memorize these. I'm simply not expecting you to memorize these. What I do want you to recognize and what I want you to learn from this is that these molecules are they're a part of the bilayer. And so what that means is that they serve different functions, or sorry, they have a functional role. So if we imagine that this is our lipid bilayer, let's just go ahead and imagine that there's a lipid bilayer. Okay. Now, when you talk about a lipid bilayer, there is what's known as the outer leaflet and the inner leaflet. The outer leaflet is what is kind of on the outside. So if we're thinking about a plasma membrane, well, the outer leaflet would be the part that's, or sorry, let me rephrase that. If we're thinking about a plasma membrane, the inner leaflet, which I will label as IL, is in contact or contacts the cytosol. We're talking about on the inside, hence inner leaflet. Outer leaflet, on the other hand, is what's in contact with everything outside of that cell. Now, with that in mind, this is important because there are certain instances, physiological responses, where the abundance of different glycerophospholipids are higher on the outer leaflet than on the inner leaflet. So, for instance, if we just, to make this simple, I'm going to say S for serine. Well, that serine, if that serine is displayed on the outside of the cell, that might be the cell communicating like, hey, I'm under distress, or um, there's something impeding my growth and my function. So I'm just trying to communicate to everything outside of me. Um, yeah, so that's one of the things that they do. And in addition to that, they can also be beneficial for like receptor proteins. So a receptor molecule might have um, it might be involved in communicating with adjacent cells. Like, hey, this is a cell that's been attacked by a pathogen. Um, I'm just letting other cells know that this pathogen is present. So maybe you want to make some reactive oxygen species in preparation for that. So with that in mind, these the things that are on the inner leaflet and the outer leaflet, they will change. And we'll get to that when we talk about membranes a little bit more. So what I want you to understand about this is that all of these molecules have different chemical properties and they communicate different things to either interior of the cell or exterior of the cell. Now, when it comes to sphingolipids, like I said, we've got that kind of 18 carbon backbone. Okay, so here's 3, and then here's our 15 more. So there's our 18 carbon backbone. And then we've got this amination at one of those carbons. And at that point, we're going to add a fatty acid. So this is kind of the construction of a sphingolipid. So a sphingolipid, that's our general kind of catch-all term. That's one way in which something like sphingomyelin can be classified. Sphingolipids are major membrane components. They're derivatives of a C18 amino alcohol known as sphingosine, which is what we see right. Let me switch colors. I can go to this dark red is what it's called. So there's our sphingosine. Now, the base unit or the base molecule of that is known as a ceramide. So if anyone has and ceramides are something that I know that my wife has in one of her like face lotions. Ceramide, it has, I think, CeraVe, not to be, yeah. So CeraVe has ceramides in it. And so it's got a bunch of this molecule right here. Now, if we proceed from there, a sphingomyelin is just an example of a sphingolipid. Now, sphingomyelins have a head group of phosphatidylcholine or phosphocholine. So here is that head group that has been added to this phosphate that is on this 18 carbon long structure. So that's why this is, if we jump back a couple of slides and we look at the different head groups right here, with those different head groups, we've got, so those head groups vary a little bit to give you different glycerophospholipids as well as different sphingolipids. Now, There we go. Um, Sphingomyelins, that's a class of compounds that have that phosphocholine as their head group. Cerebrosides, by contrast, they have a head group that would be a singular sugar, something like glucose or galactose. Now, these different cerebrosides are pretty important for like development. Um, and these cerebrosides get metabolized in different ways. And depending on how they get metabolized, they can, sorry, let me rephrase this. There's lots of genetic disorders that are associated with degradation and differential degradation or problems in the degradation of those. For instance, Tay-Sachs disease is one that's a genetic disorder, genetic issue with cerebroside degradation. Niemann-Pick disease, there's a couple of different examples. So, with that in mind, it's important to understand like what a cerebroside is, what a sphingolipid is, and that it is a pretty critical part of membrane and communication. Sphingolipids and glycerophospholipids, thinking about their role in energy storage, I really don't think that, I don't know how much they give. I haven't looked that up. But they are not big kind of leaders or functional roles in what is it? What am I looking for? Energy storage. So when it comes to, which kind of makes sense, if those are major parts of a membrane, and you're trying to like, if imagine that you're, personify. I always like to personify a cell or personify something. If you need nutrients and you look at your plasma membrane and you say, oh, okay, well, that's made up of glycerophospholipids and sphingolipids. Those are used for communication. A membrane is used for like a barrier. Do you really want to be eating that? Do you really want to be extracting energy from that? Not really. You want to find something that isn't such a structural thing, and breaking that up. Now, these different molecules, glycerophospholipids, for example, or phospholipids in general, they get metabolized and they get broken down. Now, this slide right here, you know, this is looking at phospholipases. Now, phospholipases are classes of enzymes that they are going to work on glycerophospholipids. And this isn't metabolism for the sake of like, let's extract energy from them, but instead let's release a molecule to send some sort of message. So in addition to that, cells are continuously degrading and resynthesizing membranes. So phospholipids and sphingolipids are both kind of targets for this. Again, not for nutrient acquisition, but instead they're going to be broken down just to kind of like maintain the structural integrity or their structural integrity. So I'm going to highlight our glycerol backbone. And then I'd like to focus on phospholipase A1. That is one of our enzymes that is going to act on a glycerophospholipid. And what it's going to do is it's going to release a free fatty acid. So what's going to be left behind is, well, a hydroxyl group. And this figure, what this is kind of trying to display is this is specifically focusing on, if you look at the reactants and the products, this is looking at phospholipase A2. Now, what phospholipase A2 is going to do is it's going to cut effectively in the same place as phospholipase A1, however, on carbon number 2. And so it's going to cut and leave behind, just like phospholipase A1 will leave behind that oxygen, which can then pick up a proton from water and make that hydroxyl group right here. Phospholipase A1 and A2 would both do the same thing. Now, where it gets kind of tricky, and so this is, I'm going to highlight or just put a big box around the top half of this, because what that is saying is that whether we're cutting with phospholipase A1 or A2, the product is effectively going to be the same. The only difference is A1 cuts at carbon number 1, A2 cuts at carbon number 2. Okay, so that's the differences between those. Now I'm going to highlight, I'm going to get rid of some of these markings. I highlight something else. Now we have the kind of what sets our glycerophospholipid apart from a triacylglycerol is that phosphate group on our third carbon. And that phosphate group, well, essentially where we cut related to that phosphate group, we're either going to release a amino acid or sorry, we're either going to release a head group or we're going to release a phosphorylated head group. And what I would like you to focus in on is which one of these cuts, phospholipase C or D, is going to release just the head group. If you imagine that C is cutting right here and D is cutting right here. Well, where D is cutting is the X or your head group and that oxygen, leaving behind the phosphate. D, however, is cutting between the oxygen and the phosphorus. And what C will release is your head group with a phosphate, so your phosphorylated head group. So phospholipase C releases a phosphorylated head group, whereas D releases just the head group. Now, I want you to know where each of these different substances cut so that you can kind of respond to questions related to that. Now, what we've talked about so far when it comes to membranes is their composition as harborers of energy, not nutrients, but energy. Okay, so your triacylglycerols. Then we've talked about them as major components of membranes. Okay, so your sphingolipids and glycerophospholipids. So if we run it all the way back, and we kind of account for our different classes of lipids. I'm going to clear all this out. Drawings. We've talked about fatty acids, triacylglycerols, glycerophospholipids, and sphingolipids. Now what we need to get into are our steroids. Okay, so I remember in organic chemistry too, I learned what a steroid was, and all of a sudden I was just like, oh, this is exciting. So there are non-membrane functions of lipids, and steroids are relevant to this. They serve as signaling lipids. They can be cofactors and they can be pigments and antioxidants. So signaling lipids, eicosanoids are an example of a sphingolipid. And an eicosanoid is a 20 carbon long molecule that basically gets cut and cyclized. And depending on its cyclization and the abundance of it, it could lead to inflammation. And so if you prevent that, then you're going to kind of reduce inflammation. Steroid hormones. Well, steroid hormones act as chemical messengers. So they go from cell to cell or, from one part of the body to the other part of the body to signal, hey, this is important. Liquid cofactors. If you go on to a metabolism class, you'll learn about oxidative phosphorylation and the electron transport chain, and you will learn about ubiquinone, or as I incorrectly called it for quite some time, ubiquinone. And this is a molecule that basically helps out with transfer of electrons. In addition to that, all your pigment molecules, or many of your important pigment molecules like beta carotene, and then phytoene. These are pigment molecules that if they're expressed or if they're produced, I'm sorry. These are long lipids, so they're super hydrophobic molecules. Where they're produced, you'll see a pigment corresponding to that. Then, in addition to that, they are also involved in kind of functioning in your ability to see. So vitamin A is an essential pigment for vision, and vitamin K functions as an antioxidant. So first, let's get into cholesterol. Cholesterol is based on a core structure consisting of three six-membered rings, one five-membered ring, all fused together. And then typically, where steroids, cholesterol being a steroid, where they vary is kind of these modifications to them. Now, if you look at cholesterol you'll see this alkyl side chain, and the length of that and the difference in this polar head group that's highlighted in the lower left-hand corner of the molecule, the variations there are where you see variations in what that molecule looks like and what that molecule does. Now, cholesterol is the most common steroid in your animal and or in animals and the precursor for all other steroids in animals. So this is kind of your base unit. And when that modifies or when that changes, you produce different molecules. For instance, vitamins, vitamin D being one of them. Now, there are also compounds known as phytosterols. And from a kind of physiological perspective, these help control serum cholesterol levels. So people will take things like stigmasterol, beta cytosterol, or campisterol. And the idea there is to basically help fend off or reduce their overall blood cholesterol. Beta prostate is one example of those phytosterols. Phyto being from plants. So these are steroids that are, typically a part of plant membranes. Now, with all of that said, we've talked about different lipids and where they come from, and maybe the reasons why someone should take or someone should have a diet high in this or versus high in that. Well, phytosterols, sure, they exist in dietary supplements, but they also exist in plants. They also exist in, what's a plant? What's a plant? What's a plant? Broccoli. So if you eat broccoli, are you going to get some phytosterols? Absolutely. If you eat broccoli, are you going to get some polyunsaturated fatty acids? Yeah, you will. So that's one reason why there's an encouragement, typically eat vegetables. Of course, diet is not that trivial or simple. But when we look at steroid hormones bind to very specific receptor proteins on cell surfaces. And whenever they bind there, imagine that my hand, let's look at my forearm as being basically where a lipid bilayer is. My hand is the receptor protein that binds something like testosterone. And based on that binding of testosterone, well, my arm is going to undergo a structural change. And whenever it undergoes that structural change, then there's going to be some signaling molecule on the inside of the cell, in the inner leaflet. So that would be kind of your secondary messenger, like cyclic adenosine monophosphate, or CAMP, as some people like to know it as. So testosterone, estradiol, cortisol, aldosterone, prednisone, prednisolone. These are all examples of steroid hormones. Now, major groups are the sex hormones, and those produced by the adrenal cortex. There are also lipid-soluble vitamins, which are derived from cholesterol. Vitamins A, D, E, and K. Vitamin D being one that's pretty common for people to be familiar with. That's a vitamin that when people talk about in Central Texas, for example, it's like, well, you get an excess of vitamin D in Central Texas because of the fact that there's lots of UV light, there's lots of sun exposure, and you really, your body can produce this provided that you get that sun exposure. Because what vitamin D, functionally, what vitamin D does is it regulates calcium and phosphorus metabolism. By regulating calcium metabolism, it helps, your bone strength. In addition to that, it helps muscles because if there is too much calcium inside of a muscle, then that's going to be preventing a problem, whereas vitamin D kind of helps out with that. Now, vitamin D, well, it's actually kind of a group of molecules, and they're structurally very related to one another. The most abundant of those is vitamin D3, which I'm going to go ahead and take one right now. Now, this is the most abundant form that's in your circulatory system at a given point in time. So what I just took was vitamin D3. And if I look on the bottle, what it actually says, okay, so there it is. It says, it's tough to see. Sorry, it's reflected, and I'm sure that you have this is low resolution, and I'm in a small screen. I just took cholecalciferol. I took 50 mcg or 50. Oh, no, I don't need that. This is one of the things whenever you think about a dietary supplement, the labeling, it says MCG. Now, you know what that means, but you also know that that's not the correct notation for a quantity. MC meaning micro and G meaning grams, so micrograms. Well, UG, that mu is actually the character. Mu G is micrograms. So I just took 50 micrograms of this. Okay, so essentially, I'm doing that because it's during one of the fall months, or it's during one of the later points in the year. And so my chances of getting lots of UV light exposure are reduced. The days are shorter. Um, Basically, the Earth and the sun are in different distances from one another. So this is a part of the year that my UV light exposure is going to be a little bit depressed. And as a result, I'm expecting that this step right here is not going to be done quite as, as efficiently or as often, I guess you could say, as what I get in the summer. Okay. Now, with that in mind, well, vitamin D is a fat soluble vitamin. And what vitamin D, this cholecalciferol that I just took, it's going to be metabolized two steps. One step in the liver, one step in the kidney. So in order to get this form right here, the kind of relevant and functional form of vitamins D in my body, I still have to have proper liver and kidney health. Now, that's important. These two different steps are basically just to, I think that it's helpful to look at the names and be a little bit familiar with these. Um, so I'm going from cholecalciferol to 125 dihydroxycholecalciferol. So what I started with was this right here, 7-dehydrocholesterol. Okay, so that is cholesterol, but there's one modification to it, and that's that position number 7, where we have, and I'm going to jump back, position number 7 on one of those rings. Okay, here's cholesterol. If you look at position number 7, that's in the B ring. Well, I lack, or sorry, that would be, kind of a saturated double bond. Then if I jump forward to vitamin D here, well, if I look at 7 and 8, I've got a double bond there. Okay, so that is what is going to be targeted. Or that's part of what's going to be targeted. Now I have that double bond there in my my vitamin D version, or no, my vitamin D molecule, which is derived from cholesterol. Now, what UV light is going to do is it is going to take this molecule that has 4 rings, the 7-dehydrocholesterol, and it's going to break open this ring right here. And whenever it breaks that ring open, what's going to be left behind is what was known as my A ring right here, and then my 2 C and D rings. Those are going to be left behind. My B ring is opened up. Now, as soon as that B ring is opened up with the help of the UV light, that's when the 2 enzymes, one in my liver and one in my kidney, can act on the remaining cholecalciferol, the vitamin D3, this being vitamin D3. So basically, I took a bunch of that cholecalciferol, that now a 3-ringed molecule, and what I'm relying on and optimistic of is that my liver and my kidney will go ahead and they will add a hydroxyl group here and a hydroxyl group here on carbon number 25. Position number 1, or carbon number 1 and carbon number 25. So that's what I'd like, that's what I'm hoping that is going to happen. This 125 dihydroxycholecalciferol, however, that's going to be the form of vitamin D that's really important and functional and relevant to my body. That vitamin D form is going to go on to serve as a stimulator of different things, turn on expression of different genes, turn on and activate, proteins that will either import or export to help regulate calcium in my muscles, and well as to help um, bone fortification and everything like that. Now, with that in mind, vitamin D also it has a number of different functions, one of which is it helps it kind of supports your immune system. And that language right there, supports immune system. That's the very vague and general terminology that the dietary supplement industry uses, um, because they don't have to prove it in any form or fashion. They can say things like supports, as opposed to saying something like what it actually does. They speak very generally. This supports it. Well, what does that mean? Um, at any rate, that's just an aside, and I apologize for bringing that up. Um, with that said, though, vitamin D is a very important vitamin to consume. Now, vitamin E is a group of compounds of similar structure. The most active is one known as alpha tocopherol. It's an antioxidant, and what it does is it captures free radicals like whoo and woo. These hydroxyl radicals formed as a result of oxidation by O2 of unsaturated hydrocarbon chains in membrane phospholipids. Now, I think that, of all the vitamins that are pretty interesting, in my opinion, vitamin C and D are pretty much the most interesting because I think in part they're the most well, I don't know if I want to go as far as to say understood, but they're the most well studied. Um, and I would say that they're most well studied because there's like um it's not such a vague summary of them. Okay, now for the last few minutes, what I'm going to do is I'm going to jump over and talk about membranes. So we've talked about membranes a little bit, and I believe your share did not change there, so I'm going to change that. Okay, so membranes. Now, when it comes to a membrane, just a second, just a sec.